Thank you all so much for being here today. We are excited to have Dr. Sheng Luo, Associate Professor of Biostatistics at Duke. He came to us from Johns Hopkins in August of last year, I guess. He specializes in biostatistical methods of design and analysis of clinical trials and observational studies. He's won multiple awards in statistics and clinical science and has 72 peer-reviewed articles. He's the PI for an ongoing NIH grant for statistical methodology, and we're delighted to have Dr. Luo here to talk to us about dynamic prediction of Alzheimer's disease progression, the big data approach. So thank you.
Thank you. Thank you, Alex. It is on? On? Yeah. Okay. Thank you. Thank you, everyone, for coming. Good afternoon. So this is a joint work with my former student, Kang Lee, who is working at Merck now. So, and also this work is being supported by NINDS, the R1 grant. And feel free to stop me anytime for questions, okay? So now, what is Alzheimer's disease? The Alzheimer's disease is the neurodegenerative disorders of brain. It's the #1 leading cause of dementia. And #2 is the Parkinson disease, okay? So usually people say the other way around, not the other way around. AD leading to the dementia, not the other way, okay? So currently, there's no disease-modifying treatment for AD. And unfortunately, this is the most expensive disease in America. So put that in the dollar amount. So now in last year, there are 5.5 million Americans with AD, and with the medical cost of $259 billion. But if you put that in the GDP, I calculated that a few times, and then it's 1.3% of US GDP is spent on this disease alone, not including cancer, cardiovascular disease, and everything. So this is really devastating disease and very expensive. Now, the projection is that by 2050, 13.8 million Americans will have Alzheimer's disease, and then $750 billion will be spent in this disease in 2018 dollars. So here is the projection of the rising cost of Alzheimer's disease in national level. So now we are somewhere down here, 2018. So if the trend continue, you can see that the medical cost rise exponentially. Oh, I'm sorry. I'm sorry. I should do the poll. Well, hold on. All right. So otherwise, you guys see the results. Okay. All right. So so please test to this DCRI research 803 to this the number here. Okay. Or you can go to the website to join. Okay. So here is the question. What is the average annual healthcare cost of a person age 65 and older with Alzheimer's disease or other dementia? Okay. So 16K, 32K, 48K, and 64K. Okay. All right. Give you guys maybe 30 seconds to respond. Okay. So is there a sample size here? Okay, I think most of you got it right. So the answer is the 48. Okay, 48K. So here you can see a person with Alzheimer's disease on average spend 48K per year on medical cost. Okay, but the person with the same age spend on average about 14,000. You can see it's almost the fourfold difference in terms of medical cost spending. Okay, so now the question is, can we do better here? Okay. First of all, I'm not a clinician, but I would like to boldly declare yes. Okay, please correct me. Okay. Now, the key questions, scientific questions, are here. Okay, so here I quoted from the NIH All of Us, the research program. So Anybody know this, what this is? All of us? So the old name for that is the Precision Medicine Initiative. Okay. So and then under the new administration, I think they changed that. Now I want to quote the two statements from there. There are 7 of them, major objectives. The first one is to develop ways to measure risk for a range of diseases based on environmental exposure, genetic factors, and interaction between these two. Okay. And the second one is to discover biological markers and signals increase or decrease risk of developing common diseases. Now, as you might know, this research program will collect data from at least 1 million Americans. And then they will collect the environmental data, biological data, say in terms of clinical data, medical imaging data, genetic data, and also other related data. Okay? So they want to figure out these two objectives. Now, you can see the common word here is risk, right? So the risk is the key here, right? So how do you do this? In my opinion, the tool here is the personalized risk prediction. All right, so by personalized, I mean that you have to predict for each individual, not just in the population level, okay? Now, in Alzheimer's disease studies, so there are some common feature here, okay? Let's kind of quickly go over this. Now, the Alzheimer's disease affects multiple domains, such as the cognitions, the behavior, and quality of life of the affected individuals. And second feature is that it progresses heterogeneously in times and across domain and across individuals. So if you look at the progression trajectory, you can see a lot of variation across time and across individuals. Okay. So now, in this sense, the Alzheimer's disease always collect multimodal data. Okay, by multimodal, what I mean is that they collect clinical variables such as the neuropsychological measurements, the functional measurement, and behavioral assessment longitudinally, which means that each individual will be measured multiple times. And also there are neuroimaging data such as the MRI data, and also event history data such as the time to event. For example, time from study onset to the AD onset. Okay? And also genetic data, such as GWAS data, to better understand the disease. Now, between the cognitive normal and Alzheimer's disease, there's a so-called transitional stage, which is the mild cognitive impairment. So usually people with normal cognition, some of them will progress to MCI stage, and then in this stage, some of them will progress to Alzheimer's disease, some of them may not, right? So you want to know who is going to progress from MCI to AD and who may not, right? So in this sense, we want to investigate the effect of combining multiple variables you can measure up here, okay, in the second bullet point, to predict the conversion from MCI to AD. Okay, now here comes the next question. Okay, so because we are talking about prediction here, the question here is which predictive models or method do you use the most? Okay, logistic regression, Cox model, random forest, neural network. I guess I need to put others, right? So this list is certainly not exclusive. All right. Anybody else still doing it? Okay. All right. So it looks like most of the people are using logistic regression and Cox model, right? So I think I'm addressing the right audience here. So that's exactly I have prepared for. Okay. So so now, in terms of the predictive models, most of the predictive models you have seen in the literatures are so-called static models. Okay, so such as the logistic regression and Cox model. What do you mean by static? What it means is that once you predict, usually you cannot update your prediction. Once you get the risk, you get it. You don't update, even though that the disease progression change. the risk may not change, okay? So there are some pros and cons to this method. So the first one, in terms of pros, is that it's very simple, low computing cost, very easy to calculate. You can use SAS to do it. Now, the disadvantage is that the prediction cannot be updated in a real-time fashion. I will illustrate what that means. So here, I use this simple diagram to illustrate. Okay, so on the y-axis is the square root of CD4 count, okay, which is the measurement of your immune system. Right? So here, the time 0 is the HIV infection. Right? So I want to know when the HIV-infected individual will develop AIDS, right? So suppose underlyingly, this particular individual developed AIDS at almost year 3, right? So, okay, so, and then you measure this, the square root of CD4 count, okay? So those are these black dot here, right? So you only have about 6 measurements here, right? So longitudinally. And then assuming this, the red curve is the underlying trajectory, okay? So what you can see immediately is that your measurement is subject to measurement error, right? You don't measure the underlying truth. You measure a proxy of that, right? Okay, and then plus when the event happen, you may not have the measurement right at that point, right? So when you calculate the use the Cox model to calculate the so-called instantaneous risk, supposedly you should use this point, right? Right here at the intersection to predict your risk. But unfortunately, you don't have that, right? So what do you do? Well, in literature, usually people, there are 2-way or 3-way people do it. So one way is that, well, they just plug in the baseline, right? Or they plug in the last observation, okay? Or they fit a straight line and then to approximate the disease progression. There are some pros and cons for every single one of them. But the bottom line is that none of them is right, okay? Even though that it has been used very widely in the literature. Okay, that's why, suppose your aim is to model the effect of some time-dependent predictor, such as CD4 count, to the hazard of events such as the age onset. Okay, so now the first model you come in mind is that maybe I just figure a Cox model with the time-dependent covariate, right? So very simple to do. However, the problem is that you don't know the complete history. Okay, so as the previous diagram show, you only have a few discrete points. And the second one is that you do not observe the true value. Okay, but you only observe some contaminated version of the true value, all right, subject to the measurement error. Now, what do you do? The solution here in statistics is what we call joint model of the longitudinal and time to event data. I will not get into the detail, okay? But the intuitive idea behind this is that basically you have a 2 sub-model. One model is to use to develop the evolution of the biomarkers such as the CD4 count. And then you have another sub-model to develop your, to describe your survival outcomes, such as the time to age, okay? And then you link them together using some kind of latent structure. Right? So, however, a novel use of this joint model is to make this the personalized dynamic prediction, which I will get into next. Okay? So what is dynamic prediction? So the prediction are conducted in a real-time basis. Okay, that's the key. So that whenever a new outcome measurement are available, you can always use the latest measurement to update your prediction, so that you have the latest prognosis, okay? And then you can further target treatment, initiate the targeted treatment if necessary. Now, why is it important? Well, you can predict the patient's prognosis and make a medical decision in a real-time fashion, right? So if you see an individual in a high risk of having a certain disease, you can have some targeted prevention and targeted counseling so that this person may do some right thing to reduce the risk, right? So, and also answer very important predictive questions. For example, for a particular person, say John Smith, coming in the office, and then you want to know what is the most likely outcome trajectory in the next 6 months, right? Back to the CD4 count example. You may want to predict how the CD4 count will progress in the next 6 months, right? So if you progress or deteriorate so quickly, you might need to have some targeted therapy, okay? And another question you want to ask is that what is the risk of developing, say, AD or AIDS, right? So that you can enable the personalized prevention, treatment, and care. Okay, so the last point I want to mention is that this kind of a personalized dynamic prediction has a huge impact in optimize the clinical trial design. As we all know, running clinical trial is very expensive in the US. On average, it takes about 10 years and costs about $1 billion to develop one compound and put in the market, okay? So, but in the design phase, when you enroll individuals, right, if you can predict in your target population who has the highest risk of having the event of interest, right, or has the fastest progression, you are more likely to enroll the person with higher risk, right? So you are more likely to have your treatment efficacy shown, okay? Now, so we have used the dynamic prediction model using this longitudinal clinical data and medical imaging data such as MRI and genetic data. Okay, so before I get into some detail, let me show you our final product. Okay. So this is the web-based app. Okay, so is to do the dynamic prediction of future health trajectory and risk of Alzheimer's disease. So on the left panel, you can put in the baseline characteristic. So for example, you have an age 65 male with a 12 year of education with the one copy of the minor ApoE allele, which is the risk allele for Alzheimer's disease. And down here, you can put in the longitudinal measurement. Yeah, there's something wrong with the display, but the bottom line is that you can put in the, say, for example, 5 visits. And here you can put in the each visit the measurement for this ADACOG 13 and some other variable. There are 3 more here. It doesn't show for some reason. But let's say if you put in all the data, you can click on Go. Oh, finally. Okay, maybe. Yes, right. So here it will show you the disease progression. So these the 5 points are the point I put in on the left panel. Okay. So and then this the vertical dotted line is the time of prediction. I want to, based on the 5 observations I have, to predict what the disease progression is. So you can see this is the progression with some confidence interval. Okay. And then here, this is the risk prediction. So if you want to know the probability of this person remain AD-free by year 3, the probability is about 80%. Okay. Another way to say this is that this individual will have about 20% to develop AD by year 3. Okay. You can do the same thing for year 4 and 5 and so on. Now, this is the so-called dynamic because if you have one, say, additional visit, okay? So with some additional information, you can click on Go and then you can dynamically update the prediction. So you can see now I have one more observation, right? So from there, I can predict the trajectory, okay? And then also the risk get updated. Okay, one thing I want to point out is that this line is flat because this person did not develop AD during this time period, okay? That's why you need to predict, right? Otherwise, there's no way no point of predicting, right? Okay? Any question about this? Yes?
Are people able to take this cognitive scale test? Is it like a app or you have to see your physician?
Yeah, they have to see the physician to do it.
And are they basically taking like a cognition test or? Okay.
However, in Parkinson disease, I have seen a JAMA paper earlier this year. I think a group in Hopkins developed an app, and then they can collect a few different kind of data, such as the finger tapping, and then you can do the voice detection and something else. And then the bottom line is that by collecting this data, they can develop a score from 0 to 100. 100 means the worst, the 0 the best, right? And then they can use that to detect the PD progression. But the good thing here is that this smartphone-based data is so easy to collect. If you can really monitor the progression using the smartphone data, and then you can couple that with this kind of prediction technology, right? So you can predict the risk of having Parkinson's disease in the very near future, okay? Now, next I'm going to talk about this, the paper we published in Alzheimer and Dementia earlier this year. Okay, so now the motivating example here is that Alzheimer's disease, the neuroimage initiative, or ADNI study, which is the longitudinal observational study investigating whether serial brain imaging, clinical and neuropsychological assessment can be combined to measure the progression of AD. Now, there are a few phases, okay, to this study. The first phase is the so-called ADNI 1. So particularly, we select 379 individuals with mild cognitive impairment at baseline, and then they every 6 or 12 months, they have a follow-up visit, okay, and then they follow annually. And the second phase is so-called ADNI 2. They enroll 424 individuals with mild cognitive impairment at baseline, and then very similar design in terms of follow-up. Now, the key here is that in this study, they collect clinical data such as what I have shown in the app, okay, and also the imaging data such as MRI, and also PET imaging data, and also the GWAS data, and also some biospecimen data, which is the request require. Okay. Now, here is this very simple diagram to show you what kind of data available at which time point. So we use this data a lot, this the cognitive assessment. Okay. Now, this is the very simple table to show you the baseline characteristic of the two cohort, ADNI 1 and ADNI 2. In ADNI 1, we have 379 individuals with MCI. Among them, during the follow-up, 198 of them developed AD, and 181 did not develop AD, okay? And then in ADNI 2, there are 424 individuals. 94 of them developed AD, and then 330 did not, okay? So the follow-up time is ADNI 1 is about 3.24 years, and then this ADNI 2 is about 2.7 years, okay? And then here you have a lot of this neurocognitive measurement. I will not get into the detail. Now, but I want to direct your attention to 5 longitudinal measurements we use, okay? There are a total 33 of them. Longitudinal measurement. Now, in this paper we published in 2017, we identified 5 of them, which I listed here. Those are the neurocognitive assessment. We find out that these 5 measurements are very predictive from MCI to AD conversion. Okay, so here this is the simple longitudinal profile plot show you that how this profile look like as the time go on. Okay. Specifically, I highlighted 2 individuals. You can see there's some missing data here for this subject 406, right? And you can see this the pattern change a lot for each individual. Okay. And I plotted the same thing for the other 4 variables. Okay, and specifically, you can see this functional assessment questionnaire. Okay, you can see this huge change for these individuals. All right. Now, the question is how do we utilize the data to do the predictive models, right? So first, we try something simple. Okay, so we just fit Cox model with all these variables you can get, right? Age, gender, education, ApoE, and baseline variables for this for this, the 5 of them we identify, okay? Now, if we do that, we calculate this AUC, and also without getting into too much detail, this AUC is different from what you have calculated in logistic regression because it's the time-dependent AUC. So you have to do the integration. That's why you call it IAUC, right? So, but anyway, so the higher the better, right? So if you do the internal validation in ADNI 1, you have about 0.78 here. But if you do the internal validation of ADNI 2, which is about 0.86, right? But if you fit the model using ADNI 1 data and apply to ADNI 2, you have AUC about 0.88 using Cox model, right? So not bad, right? Now, the second thing we have done is that, well, we want to augment our model 1 by adding into some MRI data, right? Such as the volumetric MRI in hippocampus and mid-temporal region of interest, and also the PET data. Now, well, if you compare this result versus this result, right, it doesn't change much, right? Almost no change. So that means the in MRI or PET does not really help us in prediction, okay? So this is one of the finding we have in this 2017 paper. So I think the key here is that from this cohort, we see that don't waste your money to do MRI, okay, or PET. It doesn't give you that much information in terms of prediction. That's the bottom line here, okay? Now, but can we do better by using more advanced method? So we use the so-called multivariate functional principal component analysis. Again, without getting into the technical detail, basically it's a flexible so-called non-parametric approach. That means you don't have to give a very strong assumption about your functional form and how the covariate affect your result, okay? So the bottom line here is that the method can extract the trajectory pattern from multiple longitudinal variables, okay? And then eventually you get a set of MFPC scores. And then you can put this score into your model, such as Cox model, okay? So this score captures the essential information, and importantly, it captures the correlation among the longitudinal outcomes, okay? And then we select 12 of them. So that at least 95% of the variance is captured. Okay, we put that into the Cox model together with other variables we know, such as age and gender. Okay, so here is what I find out. Okay, so now our model is the model 2. Okay, so well we increased the AUC from 0.88 to 0.93. All right, so about 0.05 difference. Is that enough? Is that enough? Well, that's pretty much the best we can do in this data set, okay? If we have another data set, we might be able to do it better, okay? But think about this, that the AUC, the hard limit is 1, okay? You cannot go beyond 1, right? So we are very kind of close to 1 now. So I assume it's okay. All right, so now without giving you too much detail. So here, basically, after you fit it, you have all these coefficients, and then you have a standard error, okay? But how do we use that? So basically, we can use all these coefficients to compute a so-called prognostic index, okay? It's very similar to the prognostic index you have used in other therapeutic areas, such as the cardiovascular disease area, okay? Basically, if you have their baseline age, you have a gender, education, and ApoE, and then you have some measurement of those 5 variables, you can use our method to calculate this MFPC scores, okay? And this will all be done automatically, and then you have some coefficient to go with it, so that you calculate an index, right? So after you calculate the index, you can categorize each individual into a different risk group, okay? So into low risk, medium low, medium high, or high risk group, okay? Now, here I want to show you the Kaplan-Meier curve, okay? So now if I categorize all the individual in ADNI 1 into the 4 group and follow them and see see the risk of progressing into AD. And here, down here, this is the group with the highest risk, okay? And up here, this is the group with the lowest risk. You can see the huge dichotomy in separating all these individuals, right? So, and same story from ADNI 2, okay? Any question about this plot? All right, so another use of this, the prognostic index, is that suppose I have an individual come into the clinic, right? So now how do you use the prognostic index to help this individual, right? So now suppose here I have an 80-year-old male, right? And then with the 15 years of education, pretty well educated, right? And then with the zero number of ApoE allele, right? And then here, in this the 5 variables, you have the measurements, say at baseline, at month 6, and month 12, and month 18, okay? I recorded all the numbers. Now, we can use all this information, which is the information you put in on the left panel on the web-based app, and then you can do the calculation, okay? So the calculation, well, of course, it will be done by computer, not by you, right? So it will calculate all these scores, okay, at the different time point at the baseline, month 6, 12, 18, okay? But the bottom line here is that you can calculate this, the prognosis index, right? You can see this index change as your measurement change, right? So if you remember the previous threshold, okay, so from baseline all the way to month 12, this individual is a mid-low risk group, okay? However, from some point between month 12 and 18, this individual evolve into the mid-high risk group, okay? So, so the person needs to be monitored more carefully because this person has the increased risk of having Alzheimer's disease, okay? Now, to wrap it up, basically, we developed a novel and efficient statistical method for prognosis of progression-free survival in a mild cognitive impairment patient using multiple longitudinal markers. Okay, so we evaluate our model through internal and external validation. The good thing here is that these 5 markers we used are routinely collected in observational studies. So we can use that to enhance the prognosis accuracy of Alzheimer's disease, okay, if you properly include it into the prognostic model, okay? And then you can use this model to monitor the disease progression and identify the individual with the high risk of Alzheimer's disease, and you might want to enroll them into a clinical trial, okay? Now, we have expanded this method into other disease areas, such as the Parkinson disease. Here is the one paper we published last year. And then here is the app we developed. Idea is very similar. You can use your latest data to dynamically update your risk, okay? And then also in Huntington disease, we have done the similar things, okay? And here is the app we developed, right? Now, what is the take-home message here? From clinical standpoint, in my opinion, prevention is the key, okay? So when you identify some individual with a relative higher risk of develop Alzheimer's disease, you should give them some targeted counseling such that you can hopefully reduce the risk. Okay, so now as I said, Alzheimer's disease, I think there are quite a few strong risk factors. The number 1 risk factor is the age. Okay, so the number says that if you are age 85 and above, you have about 50% chance to get Alzheimer's disease. Okay, of course you cannot change your age, right? So that's the non-modifiable risk. And the second risk is the genetic risk, such as the ApoE allele, right? And of course you cannot change your genetic information either, right? So now, what you can do is that you can change the modifiable risk factor, such that you can change your lifestyle, right, by being active both mentally and physically. Okay, So hopefully by changing this, the modifiable risk factor, you lower the risk of Alzheimer's disease, okay? Now from statistical standpoint, what is the take-home message? Now, if you are running into a problem such that you want to model the hazard or risk of time to event, such as time to stroke or time to AD or time to age, something like that, and you have some time-dependent predictor or some kind of progression marker, okay? You want to know how this progression marker will change your hazard of event, okay? So my suggestion is that use joint model, not Cox model, even though the Cox model is easier to use, okay? So if you use SAS, there's a macro called JMFit. You can do the joint model using that, okay? Now, if you have multiple time-dependent predictors, such as in Alzheimer's disease, we have 5 of them, right? So you want to know how these 5 outcomes change your risk of having AD. Okay? My suggestion is that talk to a statistician. Okay? All right, so there's no available package to do this, and talk to one of them or me, okay? And thank you for your attention. So I'm open for questions.
I tried to keep up with you. How do you see this tool being used in a clinical setting? Would the doctor update the parameters at the clinic visit, or would she go back to her office and do it later?
Okay, so this is just a demonstration. I think there are still a lot of work we have to do. If, number one, the risk prediction is relevant in clinical setting and is good to use in clinical setting. By saying that, what I mean is that we have to validate these models using more data. And then, if the model is valid enough, we need to package that up, right? So we have to make a more user-friendly platform. And ideally, this platform can be integrated into the current EHR system, right? So that you don't have to manually put in the data, and then automatically the data will be integrated into the input, right? So that when you click the button, you get the prediction. Right? That will be the next step. So yeah.
Thanks, Sheng. That was a great talk. So I'm curious about the diagnosis of Alzheimer's, and I have a couple of questions about that. One is, I have the impression that there's a lot of error in diagnosing dementia, and that there's even more error in specifying that it's Alzheimer's related. Was it critical for your underlying problem that you focused on Alzheimer's specifically, or would it have been adequate to model progression to dementia? That's one question. And then the other question is, I also have the impression that a lot of the factors that go into a diagnosis of dementia were included in your longitudinal model. And so I'm wondering if that's true, what extra information was used to call it an event of progression? Oh, I just changed. and how does that affect your models?
Okay, I think the first question you raised is about the diagnosis of Alzheimer's disease, right? So what if you misdiagnose, right? The people you diagnose as the AD, but actually they don't have AD, right? So it could happen. So in statistics, that's what we call misclassification problem, right? So I mean, there's some technique to address these issues. In this model, we did not take that into account, okay? I think the second question you asked is about the when you have these longitudinal measurements, right? Some of them will be used by doctor to do the AD diagnosis, right? So I think this is the critical point, because those are the what we call progression marker, right? So just because the doctor used this marker to make the diagnosed, that's the strong indication that this is a good marker, right? Otherwise, you may not use them. So that's why we need to use them to do the prediction. Okay, and also we selected these 5 markers from 33 of them. And then these 5, the good thing is that can be readily measured in clinics, and you don't need to have any fancy machines such as MRI to measure this. It's relatively low cost to measure these 5 variables. Okay. Yeah.
developed and presented, there was an event you're predicting. Are there ways to change the approach? Are there ways to change the approach if you want to predict like a score or a range? Or is it always kind of a binary event that you have to predict with these techniques?
Well, I think that this is pretty flexible in the sense that you can define any event of interest. In this particular case, it's the AD diagonals, right? So, but let's say if you want to, as you said, you want to see the risk of going into a range of value, right? So it can certainly be done, right? Yeah.
Thank you.
So thanks. Yeah, it's the dynamic modeling I think is so important. So thanks for that great presentation.
Thank you.
So just so I'm clear, are you saying like if you had all the information about one of these patients at one time and did a static model, would you be more accurate at predicting if you had available prior information to that one time?
Yes, thank you very much for asking this. This is really to the core of this model. The short answer is that the more information you have, the better the prediction is, right? So now, in the extreme case, let's say you don't give me any information, right? I have absolutely no idea the age, gender, ApoE, and any of these the markers information. What I can give you is the population average risk, right? So, but however, the more information you give me, the better the prediction is. And then in statistical jargon, the shorter the confidence interval is, right? So you have a more precise prediction.
But even prior, so let's say you have everything that you need at one time point. If you have information prior to that, it improves your prediction.
Yes,
Yeah.
yes. You can also take the prior information into account by using this measurement at different time point. Yeah. Hi, Michael.
So, since Chris stole my question, let's push it a little farther. Can you, because in my experience, the prior information doesn't add that much, right? You, like in the plot you have the time when you draw the line.
Yeah.
Right, perfect. Yeah, so we have the vertical line, and then we have the point that is on the line where you start your horizon, right? If I didn't have the 4 points to the left, but just the information at that point, how much would I lose in, say, the C-statistic or in any measure?
Right. So thank you for asking this. So I think, again, the more prior information or the more longitudinal information you have, the sharper the confidence interval is, okay? So ideally, you should have a more measurement, right? But I know that in this case, that you cannot, right? Because you have to go to the clinic to measure, right? But in the Parkinson disease example, if you can use smartphone to collect data, right? You can easily have maybe daily data, right? So with this, the so-called intensive longitudinal data, you can have a very precise prediction. So that's the beauty of it.
Okay, so you gain on precision rather than discrimination accuracy by having it.
Yes, right. So you're absolutely right about this. When we present this IAUC, this is about discrimination, right? So you can see I only gain about 0.05, right? So another thing I did not show is that I don't want to show too much mathematical detail. It's about bias, right, or calibration. Okay, so that part we can do much better than the Cox model. Okay, yeah, yeah.
I have a question about the threshold, and I saw the threshold ranges from negative to the positive. So I think it's specific for the disease, or it's a general rule, because I predicted loss to follow probability in spinal cord injury patients, that I might consider like this model to predict a better follow-up rate. So I'm curious about that one.
Yes, thank you for asking this. So these thresholds are data-driven, to put it in a simple form. So it's data-driven by this particular data set. If you change the data set, number one, your model might change, your coefficient might change, right? And the threshold also might change. Those numbers are from the quantile. Okay, so I look at 25th percentile, 50th percentile, and so on. That's how I selected this cut point. Okay, so of course this is again data-driven. Okay, yeah, thank you. Yeah.
Have you built a prediction model yet for how much this could save in public health spending?
Well, I mean, this is the billion-dollar question, right? So, yeah, you can see the high rising costs, right? Well, I guess the point I try to make is that prevention is the key. Okay, so before they develop AD, you should do everything you can to prevent that. Unfortunately, if you have AD, I mean, there's not much you can do from medical standpoint to help. So there's no any available treatment to slow down the progression, not to mention cure. So that's the, well, yeah. Okay. Any other question? Thank you very much. Thank you for coming.